Well, Masad, thank you very much for this very kind introduction. It's a great honor for me to receive this first OTS Lifetime Achievement Award, which I accept with great pleasure and great appreciation. It's wonderful that this work of my group is recognized in such a way. After all, the group laid the foundations for what's going on even today here, many, many years ago, without realizing in which way it would finally develop. Now, I thank the OTS for that. Now, besides this sort of recognition of our scientific work, I would add a somewhat more personal comment, and that is, I take it as a sign of being a member, if that's the right word, of the nucleic acid chemistry group or family, where I have been sort of associated with for many, many years. Actually, you can count it in decades. And it was always, and is still, very lovely to be welcome in this group and to be able to discuss science, but also beyond the science, the personal interaction with so many of colleagues and friends, which I'm very much delighted about and which I appreciate very much. So now I will turn to the science, and I will give you a slight historical overview, and I will not talk about oligonucleotides, because all of you are very familiar with phosphorothioate oligonucleotides, much better than I am actually nowadays. But I will give you sort of a few examples to demonstrate in which way phosphates differ from phosphorothioates, which is very often sort of underestimated. So it will be the few examples I had to select because time is short, and I only could demonstrate a few of them, and I apologize to those of you whose work I cannot mention in this brief presentation. So let me go back to, or not go back, but go to the beginning, and that is where it all started. I first synthesized adenosine nucleoside phosphorothioate for no particular reason. It was just out of curiosity, because at that time, lots and lots of modifications had been done on the bases and the sugar, and I thought, as it turned out wrongly, there could not be any more be done in this sort of area. But the phosphates had hardly been tackled, with one exception. So I just wanted to see whether, and if so, how the phosphorothioate would differ from the phosphate. And as indicated here, where is this here? This one. You see that there is no reaction. And Manu mentioned that paper in his talk on Sunday morning. This was sort of viewed rather critically by the scientific community at the time. I remember a dinner I had in Göttingen with Gobind Khurana and other well-known nucleic acid chemists, and they were not quite sure whether this would be of any importance whatsoever. But somehow I stuck it out, surprisingly. And so we thought whether this sort of resistance would apply to what is more interesting, the phosphorothioate diesters. And the simplest one to synthesize was this sort of uridine nucleoside phosphorothioate. And then you came across for the first time for a chiral phosphate, phosphorus, and then you have the two diastereomers. And how do you separate these things? Well, like so often, we were rather lucky in the sense that one of them crystallized, and Wolfram Sanger did the structural analysis of this and determined this configuration, which then is the RP isomer. And as it turns out, only this one is a substrate for pancreatic ribonuclease. So I thought there was a little extrapolation from here to there. And I will come back to that in a minute, because it's at the basis for a whole field of development of the phosphorothioates. And we then went on to what we thought was really more important, is these sort of dinucleoside phosphorothioates. And Peter Berger synthesized these two UPSAs, and we separate by a method which I can't remember anymore, the two diastereomers. And you can see that there is a difference in the reaction with pancreatic ribonuclease and the snake venom phosphodiesterase. And when you look at it, the RP diastereomer is a considerably better substrate than the SP. But even this is a poorer substrate than the unmodified one. Now, this is sort of a quick overview, which I don't want to go into detail, but I just want to show you here this compound I mentioned in the last slide, and that is a substrate for the pancreatic ribonuclease. And it was the first one in which it was shown that you can use these diastereomeric phosphorothioate substrates to investigate the stereochemistry of enzymatic reactions and in which way they sort of transfer nucleotides to water or to another nucleotide. And this work was done with David Usher by a somewhat different method from the one which I did with Wolfram Sänger, and it turned out that this was sort of an inversion of configuration. Don't get confused by they're both RP, but that has something to do with how you determine the nomenclature for that. But the inversion of configuration at the phosphorus, that's indicative of an SN2 mechanism. In other words, there is no covalent enzyme intermediate. And this has started an enormous amount of stereochemical analysis of these type of enzymes. And I think by now there must be 50 or 60 enzymes have been investigated. And 90%, if not more, go by this, what I would call a simple mechanism, the SN2 mechanism. And this was done by many groups like Jeremy Knowles, Perry Fry, Steve Benkovich, just to mention a few. And what's important here, I also show you that other nucleases, where this SP is a diastereomer for these two, but not the RP, and but the RP is a substrate for the snake venom phosphodiesterase. So you can identify the phosphorothioate configuration by using different phosphodiesterases. But important here is actually that the alpha thiophosphothioate, which is also exists in two diastereomers, only the SP diastereomer is a substrate for all the DNA or RNA polymerases we know to date, of at least for which it has been determined. So when you make polynucleotides by the enzyme, you get the RP configuration, and there is no enzyme around yet where you get the opposite, where you would get the SP diastereomer. Now, we then use this sort of RNA polymerase to make a polynucleotide, and this was early days, and we could only do the poly-RAU, where we replaced the UTP by the UTP alpha S. So every other internucleotidic linkage is the phosphorothioate, and that is of the RP configuration. So we wondered what the degradation would be like, and as you see here, this is the degradation with neogismal phosphodiesterase of the unmodified one, and this has every other which is a phosphorothioate, and that obviously is much more poorly degraded, even though it has the RP diastereomer. Now, at that time, Tom Merrigan and his postdoc, Eric de Klerk, you might know one or the other, they were interested in looking at the induction of interferon with polynucleotides like poly-RIC. And Tom thought one could possibly improve it by stabilizing the polynucleotide. And he talked to one of his colleagues next door, and this was Arthur Kornberg, and he said, why don't you turn to Fritz Eckstein? Maybe he can show you how you do it with phosphorothioates. And then that's what we actually did together then. I mean, what we did was we prepared the polynucleotide, and by that time we could actually do it also with the ATP alpha S, and not only with the UTP alpha S. And then we had this poly-RAU, which was fully phosphorothioated. And indeed, Tom and Eric could show that it's most stable in serum, and it introduces more interferon, and it shows much better viral resistance than the unmodified. And a similar effect we could show, but not to such an extent with the poly-IRC. And this is actually the sort of experiment which sort of the basis for the development you are all sort of in terms for which extended to the polynucleotides. Now, I show you the people who you have to be thankful to, and this is Tom Merrigan, and this is Eric de Klerk. He's retired now, and he is retired now. And but I should show you that those are the ones who really did the important work. The first time these phosphorothioates were tested in some biological systems rather than my in vitro with isolated proteins. Now, so this was then the beginning of phosphorothioates becoming more popular, and Art Krieg in 1995, he introduced the here's Art Krieg. He introduced the phosphorothioates in his CpG oligonucleotides, and they have been in this sort of system forever, basically, and even in the sort of spherical oligonucleotides which have been published recently. That's also the phosphorothioates. Now, this is something I'm really reluctant to show because you all know about this in great detail, but I just want to mention it. And you know, this is sort of ISIS work with the gamma and phosphorothioates all the way through. And I would like to mention several points here. And one of them is that it was Jerry Sonn and Wojtek Stets who at that time, or at some time, synthesized polynucleotides chemically with phosphorothioates at each step. So more or less the sort of further development of the Marv-Guthers technology. And that way one could synthesize larger oligonucleotides with phosphorothioates all over. We had done it only sort of with 1 or 2 phosphorothioates in an oligonucleotide chemically by hand. Now, the introduction of the phosphorothioates into oligonucleotides, whatever, the reason is that the nuclease resistance to a certain extent anyway. But what we have learned over the years by ISIS work and others, the phosphorothioates have different effects. They facilitate cellular uptake, and they also interact more readily with proteins and form tighter complexes with certain proteins anyway, which is sort of important. Now, I want to address the phenomenon of resistance, and actually Marv-Guthers has shown the slides. I'm going to, Marv-Guthers. Mano, I confuse you with Marv. I apologize to Marv, not to you. And so this goes back to something which we saw rather early, that when you put in the phosphothioate, bloody hell. that you put the phosphorothioate at the 3' end. That is then not removed by the kleno 3', 5' nuclease, but it differs for the two diastereomers, probably not unexpectedly, and the SP is totally resistant, and the RP is cleaved readily, but somewhat more slowly than the normal one. So it was really the work of Tom Check and his graduate student Bräutigam, who figured out why that was. And they have the crystal structure of this oligonucleotide with the normal substrate and with the RP and with the SP. So this is the normal the normal picture, and the important part are these two metal ions, and you are all familiar with the Steitz's sort of description of how two metal ions are important for catalysis. And then this here is also the RP diester. You see one of the oxygens here is replaced by a sulfur, and that is readily cleaved more or less like the phosphate terminus. But when you go to the next one, you see, and that would be the SP, the sulfur is here, and it sort of dislocates or even loses out the metal ion on this position, which then also removes that metal ion here. And that is because of the larger size of the sulfur, which is the van der Waals radius is about 30% larger. And this, what I would consider a small change in size, leads to the dislocation of the functional groups at the active site of this particular enzyme. Now, this is the only example where we really know in detail, structurally, why there is the differentiation between the two diastereoisomers. But of course, it's not very difficult to imagine that instead of two metal ions, like here you have two functional groups of an enzyme. You have acid or bases, you have carboxyl groups, or you have amino groups, or something like that. And they could also be sort of so dislocated that you get inactivity. There is another sort of explanation was put forward, and I think it was Steve Benkovich a long time ago, and he thought that it is in the transition state that the phosphorothioates cannot be accommodated as well as the phosphates. So, but the argument is basically the same as here. So I want to show you sort of some other properties of phosphorothioates which are not so interesting for the oligonucleotides or the therapeutic application of these phosphorothioates. But I think it should give you a wider picture of the phosphorothioates and the differences in, well, let's call it the chemistry. And one of them goes back to Mildred Cohn. And Mildred Cohn, just to remind you something which you probably don't know, is she was the first person to get the phosphorus NMR of ATP, which was an enormous step forward in those early days. And she had the idea coming based on Pearson's work in science that there are the weak and strong acid and bases, and she said so the weak sulfur should interact with weak metal ion like manganese or cadmium or zinc, whereas the oxygen, hard acid base should interact with magnesium. And she did some examples, and it turned out to be true. And that has been applied in the Czech group in various ways. Certainly in the group 1 intron, where the intron is removed. I won't call it splicing, but I mean, it's sort of similar to that. This is magnesium dependent. And so, where are the magnesium ions positioned to really be essential for this activity? So, in various steps, various phosphates were sort of changed into phosphorothioates here and there, in each one separately. And once you have a phosphorothioate there, you don't get activity anymore because it can't really interact with magnesium anymore. But with manganese, then you get activity again. So they could identify the two magnesium ions, metal ions at this position interacting in this sort of particular way, and this metal ion in this particular way. This one, of course, could not be identified, but the extra structural analysis, which was done later, actually certainly confirmed the positioning of these two metal ions, which were found by this sort of biochemical analysis. So that's certainly, and that has been done for many enzymes and ribozymes to identify metal ion positioning. Now, and there is another property of the phosphothioates, and that is that they're nucleophiles. So that they can interact in many ways with other groups. And here is sort of the internucleotidic linkage to the phosphorothioate, and it can react with iodine. And that, without going into the details, with that iodine, the sulfur can be removed, or you get removed, and then you get cleavage. And this is shown here for RNA or for dinucleoside phosphothioates. And in this way, you can get sequencing of RNA. And that is shown in the next slide. And this is done by Jerry Gish. And so here, this is a particular tRNA, and that is prepared by exchanging each nucleoside triphosphate by the phosphorothioate in 4 different experiments. And then you run a gel, and then you can actually read the sequence. And this can also be applied to DNA in a slightly different experimental setup. So you have to replace the 2' hydroxyl. Water does a good way in that. And I will come back to that in a minute, I think. And that's another sort of an example of nucleophilicity. That means once you have a phosphothioate diester, you can attach all sorts of groups. I mean, for autoactivation, and that has found the right application. And you can actually, the ATP gamma S can actually transfer the phosphorothioates to proteins, and once it's attached to the protein, you can then attach something to the phosphorothioate, like sort of label, which you can detect with an antibody. In this way, you can identify which kinase sort of interacts with protein. Now that's shown in the next slide, but I skipped that. That would just go too far, I think. Now, as it turns out, maybe a few years ago, I don't know how many years it was, Nature caught up with us. Nature finally realized that it's a good thing to introduce phosphorothioates into DNA. And I show this in this slide here. So it's work by a Chinese group, Wang, and then it has been in collaboration with Petit at the MIT. And it is, they found that in a large number of bacteria, I have forgotten the number now, but it's several hundred, there are short stretches of nucleotides, sort of tetranucleotides, hexanucleotides, which contain a phosphorothioate. And they contain a phosphorothioate in the, I don't have that here, do I? Oh yeah, here it is. They have it in the RP configuration. And my first thought was, oh, well, that must be being introduced by polymerase, because, I mean, as I said earlier, that produces the RP. But that's wrong, because that's done post-replicatively. So there is a phosphate first, and that's turned into phosphorothioate. And if anybody of you looks for what to do in the future, one, I think, would be a very interesting problem to solve is, what is the enzymatic mechanism for this? One knows that it's a DND gene family which contains about 5 enzymes. They in some way change the phosphate to a phosphorothioate. The groups which now work on this, they're really more interested in the function of what sort of effect this substitution has, rather than how it comes about. Now, the function of this is sort of unclear, because it looks as if part of it might be part of a restriction modification system. So the 10% or 20% of the bacteria in the mouse gut are contain this phosphorothioate, but not all the consensus sequences are phosphorothioated. And there is also phosphorothioate in bacteria which don't have a restriction enzyme. So the idea is that there is much more to it, and nobody knows yet what it's good for, rather than being part of, possibly, of a restriction in the nuclease system. So I think this is the last sort of science slide I show, and this is another one coming up, which just shows you that I was not the only one starting off working in the phosphorothioate field, but there are students and postdocs up here, and there are important collaborations. I don't want to go to the names because there are just too many of them. And then, of course, there are colleagues who expanded the scope of the phosphorothioates where I had nothing to do with them. I mean, this has become a field which sort of moves on its own. I mean, with you, not just on its own. And so it was unpredicted when we first did the first sort of experiments that this would be expanded into something that you could gather a conference with 460 people. This was unheard of, but that's the way it developed, and I think that's, well, I'm not proud of it, but I think I enjoy that it sort of developed in this particular way. And I certainly like to thank the OTS very much for recognizing this work in this particular way. Thank you very much.